Morgan expanded on his work with the mutant and wild type phenotypes. He began to look at the offspring that would be generated from the different breeding experiments. When he looked at the F1s, the F2s, and so on, he noticed the patterns. He noticed that some of the offspring phenotypically, remember phenotype being the physical, resembled the parents. Others would be a mix of the 2 or 3, phenotype, mutant and wild type phenotypes that they would be tracing. The ones that look like the parentals, he referred to the parental types. The ones that were a mix were the recombinants, or the recombinant phenotypes. Remember, in the last lecture, these would have been the offspring that were tan-bodied vestigial wings, black body, wild-type wings, a mix of the two parents. What Morgan's lab also figured out was the frequency of recombination. Flip the coin, heads, tails, 50-50. All of Mendel's probabilities were based on the 50-50. Which way were the chromosomes going to segregate? How one chromosome segregates has nothing to do with the others. So since it's a coin toss, all probabilities in Mendelian genetics are 50-50. What Morgan's lab found, what they figured out, is that not everything's 50-50. When you have genes, phenotypes that are linked because they're on the same chromosome, the frequency, the probabilities are going to be less than 50%. 50-50 coin toss, Mendel. Anything less than 50-50, then you need to start looking at whether or not the genes are linked. As we saw in the last lecture in, what was it? Or the last slide in lecture D. Not everything is perfect, okay? You're not going to get just the parentals, and you're not going to get, what are referred to the recombinants at, the same frequency every time with every different wild type, and mutant crossbreedings you do. The ratios would be different. Now, every time they did the black, the wild type crossed with the black vestigial wings, the frequency, of the ratios would be relatively the same. But when they tried different combinations of mutant phenotypes, the frequency, the offspring frequency, would be different. So what they were able to figure out is basically a way to discuss that crossing over when the tetrad in prophase 1 gets entangled. Again, does it always happen? No. Is there a probability of it always happening? Yes. So they're able to figure it out. During the F1, you have the heterozygous crossing with the homozygous because of our test cross. F1's from the heterozygous here is from the F1 being crossed with the double recessive. Here is our tetrad during meiosis 1. Tetrad, meiosis 1. Specifically in prophase 1. Exiting out of meiosis 1 into meiosis 2, you have the separation of the homologous pairs. But because of entanglement, notice this. B plus VG plus B plus VG. BVG plus BVG. As I talked about in the last lecture, here are your 4 different combinations of potential gametes. Even though they are linked and on the same chromosome, if they were linked and you did not have this crossing over event, half of them would be, half the offspring would be B plus VG plus, the other half would be BVG. But because of this recombination, this crossing over, you get these recombinants right here. Red with pink tip, pink with red tip. All red, all pink. These are the parental, these are the recombinant. But look at the numbers. 200-ish, 1,000-ish, 5 to 1. Crossing over can occur at all times, but it will not occur all the time. What they figured out is that you can then calculate a frequency. Add up 206 plus 185 gives you 391 recombinants. Add up the total, 2,300. Yes, during this one experiment, somebody had to sit down and count 23 fruit flies and record body color, wing type. Do the math, 17%. What does 17% mean? Well, it means it's a 17% chance that if you were to repeat this breeding, 17% chance of the offspring being the recombinant. 17% of the offspring that will be generated from that breeding, that crossing, will be recombinant. Here you look at it a little bit closer. Not going to worry about it. So you can see B plus VG plus, BVG, and then the 2 after the crossing over, the recombinants. You go through and you start seeing heterozygous, heterozygous, homozygous, homozygous, and then some weird things, something in between, something in between. 391 divided by 2,300, 17% chance of this